Global warming is pushing hundreds of new ticks and mosquito species to new climatic areas. And with them, also a number of human pathogens are making their way into colder climatic areas. These viruses belong to families like orthoflaviviruses and include important viruses like dengue virus, West Nile virus, or tick-borne encephalitis virus. Unfortunately, there are currently no antiviral therapies on the market, and therefore we should improve our basic research efforts. Viruses are microscopic thieves. They need to steal certain components from the infected cells to replicate efficiently. So if we now learn and identify what are the cellular components, we have a unique opportunity to develop novel antiviral strategies that target such components and have potential for broad-spectrum activity. In our study, we focused on one viral protein that is called the non-structured protein 4B. And this is an integral protein that sits at the heart of the replication machinery and for which very little is known about its function and its partners. Using the latest development in mass spectrometry and advanced analytical technique, we were able for the first time to map hundreds of cellular proteins that are bound by the NS4B protein of different pathogenic viruses. Importantly, we discovered that one of such components, called UBA5, is particularly required for replication of Zika virus, and we discovered that this is an integral component of a cellular pathway called UFMylation. This pathway is involved in a variety of different cellular functions ranging from cancer development to DNA stress and neurological development defects. We discovered that by repurposing small molecules that target, that inhibit this pathway, we can actually achieve potent antiviral activity against Zika virus. This does not just apply to cells, cultured cells, but also turns true in animal models of infections. As part of a large collaborative effort with researchers in Singapore and in Canada, we were able to show that small molecule inhibitors targeting this pathway are able to inhibit efficiently Zika virus replication also in animal models of infection, increasing also their clinical outcome. Importantly, in our study, we discovered that these small molecule inhibitors are not just effective against Zika virus, but they actually exert a broad spectrum antiviral activity against multiple members of this virus family, including Japanese encephalitis virus, yellow fever, or West Nile virus, just to name a few. We are currently developing this compound further as the next generation of host-targeting antivirals. In summary, in our study, we were able to identify and map for the first time hundreds of cellular components that are bound by the NS4B of important pathogenic orthoflaviviruses, and we discovered a critical role for one of this cellular pathway called UFMylation in viral replication. Most importantly, we discovered that we can pharmacologically target this pathway to achieve a profound and broad-spectrum antiviral activity. We are currently developing these compounds further as the next generation of broad-spectrum antiviral inhibitors.